Hi, everyone. Happy Friday. I'm Tanya Stevenson, the CEO of the Myotonic Dystrophy Foundation. I am delighted to welcome you to our Meet the Drug Developer session for myotonic dystrophy today. It's our great pleasure to introduce and to host Dyne Therapeutics. Following a short intro by MDF, Dyne will present on their progress of the development of their therapeutic for myotonic dystrophy. Then we're going to moderate a few questions after the presentation. So please do send your questions in during real time when the presenters are speaking. A little bit about MDF. We envision a world with treatments and a cure for myotonic dystrophy. Our mission is community care and a cure. We support and connect the myotonic dystrophy community. We provide resources and advocate for care. We accelerate research towards treatments and a cure. MDF provides a number of resources and support for our families and for healthcare providers. And this includes toolkits and publications, including handbooks and clinical care recommendations, support programs with about 30 support groups. We welcome people from across the globe. These support groups are held in 3 different languages, most in virtual settings, so you can participate from the comfort of your own home. All of our events and activities, including those support groups, can be found on the calendar of events on our website. Our Digital Academy is our library of past presentations and videos, including past conference sessions. They're all hosted on our website and available to the public. Today's session will be hosted and posted to the Digital Academy probably sometime next week. We will send out an announcement about its availability. We hope that you save and already have in your calendar May 2nd through 4th for the 2025 MDF Annual Conference in Indianapolis, Indiana. We hope to open registration within the next month to the international community. You can look for updates about the conference and registration itself at myotonic.org. July is Myotonic Dystrophy in Motion Awareness Month. This is an exciting time for the community with networking events and interactive sessions. We are currently now in week 2 of this inaugural event. We have 3 more activities. You can sign up for them at myotonic.org/in-motion-24. We have Little Things Count exercises for everyday life, the natural world, which is looking at the benefits of nature and breath work, and then a virtual Zumba class on the last day of the month. We are also picking up our Meet the DM Drug Developer series, as you see here today, with special efforts and focus to hear, especially from programs in or entering the clinical trial phase. So we will hear from Dyne Therapeutics next week, next Friday. If you haven't signed up, it's not too late. And from PepGen in early August. So please do sign up in advance and submit your questions so that they can build answers to those questions into their presentations. For MDF, our drug discovery efforts in general in the field, and especially those entering the clinical trial phases, are a culmination of our mission and helps us to hold on to hope that change is very near. Testing of new drugs in participants under the oversight of international regulatory agencies objectifies this hope and makes it real and tangible and exciting. Looking at our drug development pipeline helps illuminate the tremendous progress that's been made just over the last couple of years. We now have dozens of companies focused on myotonic dystrophy, and 9 are in the clinical testing phase. Right. Clinical trial activities tend to fall into 4 broad areas and follow the central dogma of biology. From left to right, we have DNA harboring the gene in DM1 with the expanded repeat. In a process called transcription, the RNA is made, and RNAs are ultimately used in a process called translation to make function proteins. In myotonic dystrophy, the RNA harbors the expanded repeat and forms highly organized structures that ultimately function as a trap for important regulatory proteins. We often refer to these traps as hairpin loops. When these regulatory proteins are not available because they're trapped, cell and tissue and organs can no longer function properly. So drug discovery efforts tend to target in 4 broad areas. In one, efforts focus on the repeat in the DNA using inhibition of transcription or gene editing approaches. In 2, they aim to inactivate the toxic RNA repeat. In 3, they try to compensate for those trapped proteins. Or in 4, they try to correct the downstream cellular effects. Today, you're going to hear from Dyne, who will talk a little bit about the inactivation of the toxic RNA approach that they are utilizing. So without further ado, it's our great pleasure to introduce our Dyne speakers for today, Dr. Ash Dugar and Ms. Catherine Beaverson. Dr. Dugar is the Chief Medical Affairs Officer at Dyne. He brings to Dyne over 20 years of broad experience across medical affairs, clinical and commercial development, and real-world evidence generation experience. Prior to joining Dyne, he served as the Vice President and Global Head of Medical Affairs at Dyne Therapeutics, Intracellular Therapies, Roche, and Pfizer. He began his career at the National Institutes of Health. He completed a Pharmacoeconomics and Outcomes Research Fellowship at the University of Michigan School of Public Health and earned his MBA from Pennsylvania State University and his PhD in pharmacology from the Pennsylvania State University College of Medicine. Also with us today is Ms. Catherine Beaverson. She is the Vice President of Global Patient Advocacy at Dyne. She's guiding the engagement with and implementation of collaborations with patient advocacy groups to integrate patient experience and expertise into the medicine's development and delivery continuum. She's held similar roles in industry with Pfizer, Boehringer Ingelheim, and Amicus Therapeutics. A professionally trained genetic counselor, she spent 10 years at both New York Hospital, Weill Cornell Medical Center, and Memorial Sloan Kettering Cancer Center. Ms. Beaverson received her bachelor's at Swarthmore College and her master's of science in human genetics from Sarah Lawrence College. Many of you have submitted questions in advance when you registered. Thank you very much for that. You may also submit questions while speakers are presenting today, and they'll do their best to answer questions during our Q&A session at the end of the presentation. So you can submit your questions using that Q&A function, typically found at the bottom of your Zoom screen. A window will pop up, and you can type your question in directly, and it will go to our panelists. Okay, it is my pleasure to introduce the Dyne Therapeutics team. Welcome.
Great. Thank you, Tanya. So my name is Ash Dugar, as Tanya said, and I'm the Chief Medical Affairs Officer at Dyne Therapeutics. First off, we'd like to thank Tanya and the entire MDF organization for the opportunity to present our DM1 program. It's been really wonderful talking to the community over the years, and it's always a privilege to speak to the myotonic dystrophy community. And I want to thank you for your time and attention today. And with me is the head of patient advocacy, Catherine Beaverson. I'll gush for a second, and then if Catherine wants to introduce herself. But Catherine has been part of the neuromuscular advocacy community for over a decade, and has been an amazing partner with me in this journey at Dyne. So I'm really so happy that we get to work together. Catherine?
Thank you, Ash. And I won't go through my introductions, but just will extend an additional thank you to the MDF for always being so gracious to allow us to speak with the community, engage with the community, and offer such terrific platforms such as this. So thank you.
All right, so we'll get into it. And again, please send your questions as we go through the presentation here. That'll be very helpful. Next slide, please. I'd like to remind everyone that we'll be making forward-looking statements today, and you can refer to our website for more information. Next slide, please. So you've seen me present this slide a number of times, and it's really, really important to us. At Dyne, it all starts with our mission to deliver life-transforming therapies for people living with serious muscle diseases, with myotonic dystrophy type 1 being a priority. Our mission is woven deeply into our core values, and this inspires our employees each and every day. Next slide, please. So I'm going to do a brief reminder of our FORCE platform, but just before that, I wanted to emphasize there are essentially three key challenges as to why antisense oligonucleotides, ASOs, which are essentially the payload that is going to the nucleus of the cell to correct the genetic basis of disease. There are three main reasons why they have significant limitations in the past. The first is being able to get the antisense oligonucleotide into the nucleus to be able to harness the cellular machinery. The second is targeting the delivery to muscle fibers and key organs involved in myotonic dystrophy. And the third is if you're able to get it to the nucleus and deliver it in a targeted manner with limited off-target effects, to have broad distribution, enough distribution in those muscles and organs that matter in myotonic dystrophy type 1. So the FORCE platform is our solution that we've developed to overcoming these challenges. The platform is expressly designed to efficiently and reliably deliver antisense oligonucleotides to where they need to go, to skeletal muscle, including diaphragm, cardiac muscle, and smooth muscle, to target the genetic basis of the disease. And the FORCE platform consists of three components. On the left-hand side, you can see the Fab, or the antibody fragment, or the antigen binding fragment. You can see this Y-shaped figure as the full monoclonal antibody. Well, we took the top portion of this full monoclonal antibody and engineered it to bind tightly to something called the transferrin receptor 1, or TFR1. TFR1 is highly expressed across the surface of muscle cells, including heart cells, breathing muscles, skeletal muscles, and other important areas involved in myotonic dystrophy. So the Fab targets the TFR1 and uses it to deliver the antisense oligonucleotide while maintaining the ability of TFR1 to transport iron into the cell. Because TFR1 has a natural function, which is iron transport. So we're piggybacking our ASO on to this receptor. It's still doing its normal function, and we get to use its function and deliver the ASO to the cell. And I'll illustrate this a little bit more in the next slide. On the bottom part here, we have a clinically validated linker, which we chose because of its demonstrated safety and efficacy in third-party approved products. So using a linker that's been used in other products is helpful to de-risk the platform. And then a payload, which we rationally selected to target the genetic basis of disease. And this is where we can leverage the modularity of the FORCE platform. And what I mean by that is for nuclear targeting diseases such as DM1, where we know we have to degrade the toxic DMPK in the nucleus, we chose an ASO payload. For diseases that are more cytoplasmic in nature, or where the target resides in the cytoplasm, we could choose an siRNA, for example. And we also have the potential to utilize small molecules or even enzymes with this platform. So I say all this to describe the modularity of the platform. Next slide, please. The mechanism of FORCE is different from other approaches. As I mentioned earlier, we're essentially using the cell's natural biology to deliver our investigational medicine to muscle cells and other cells. Our Fab binds tightly to TFR1. And TFR1, as I mentioned earlier, is highly expressed across the surface of muscle cells, including skeletal muscle, cardiac muscle, and smooth muscle. Now, the Fab acts as a kind of key that unlocks the TFR1 receptor and allows our ASO to enter the skeletal, cardiac, and smooth muscle and target the genetic basis of disease at the level of the nucleus. So in a way, we use a Trojan horse approach, if you will, to drive the ASO into the cell using the TFR1 receptor without disrupting its normal function of transporting iron to cells. So the FORCE platform harnesses the natural mechanism of the TFR1 receptor mediated delivery to transport that ASO across the cell membrane. And this is also differentiating because we're not using any membrane destabilizing agents to deliver the ASO into the cell and then leave the endosome, which you can see in the middle of the slide here. And by targeting muscle specifically using TFR1, we have the potential for a wide therapeutic index, resulting in less frequent dosing and chronic administration. And that'll be illustrated when I go through the study design. Next slide, please. So I know I don't have to tell this community much at all about myotonic dystrophy type 1 and the symptoms associated with DM1. So very briefly, as you all know, it's a devastating disease with no approved treatments. Tanya did a very nice description, talked about this being an autosomal dominant triplet repeat expansion disease of the DMPK gene. And in DM1, the higher than normal number of toxic CTG repeats form large hairpin loops that remain trapped in the nucleus. And you heard about these hairpin loops that are trapped from Tanya's very clear description. And this forms nuclear foci that then trap splicing proteins and lead to abnormal splicing in many genes. Now, we often get the question of what is splicing? It's essentially a cut and paste function of the body. As you know, RNA is a series of instructions. And some of those instructions are needed to make proteins and others aren't needed. So our body goes through this process of splicing to remove the non-coding pieces, the non-coding sequences called introns, and then join the coding regions known as exons to be able to make normal proteins. In DM1, abnormal splicing, or we now call this a spliciopathy, so abnormal splicing or spliciopathy, results in a wide range of symptoms that you can see on this slide, often characterized by onset of muscle myotonia, which is the inability of muscles to appropriately relax after contraction. This is followed by weakness and atrophy of skeletal muscles and symptoms, including cardiac conduction abnormalities, gastrointestinal and metabolic disturbances, respiratory distress, as well as CNS manifestations like excessive daytime sleepiness and fatigue. Next slide, please. Today, Dyne is well on its way towards achieving the promise of FORCE and delivering for patients. We've delivered exciting clinical data from our DM1 program, and I am very pleased to share our latest data readouts from this past May. Next slide, please. Importantly, our trial has been informed by multiple stakeholders. Prior to starting our Dyne 101 clinical development program, we proactively obtained expert feedback, meaning we engaged key opinion leaders or KOLs from various disciplines, including neurology, cardiology, physical medicine rehabilitation, pulmonology, and physical therapy, and other disciplines as well. Importantly, we've gathered this feedback from across the globe. And there's also a great research network that has generated a great understanding of the natural history of the disease. And they have taught us a lot as well as we think about contextualizing the results we get from our study. We've also received advice directly from patients and caregivers. They've provided insights based on their living with DM1, as well as experience with past and current trials. And this input has been critical in ensuring a patient-centric design. And it's included, but it's not limited to key considerations for choosing a clinical trial, feedback on biopsies and functional outcomes that are meaningful. Very importantly, identification of the most burdensome or difficult parts of the trial and how to mitigate that burden. And finally, how to ensure patients and families are supported throughout the process of being in the clinical trial. Next slide, please. So before I go into detail with respect to the data, I just want to briefly review the ACHIEVE study design. ACHIEVE is a global trial, and we designed it to be potentially registrational. The primary endpoint is safety and tolerability, and we are looking at many other outcomes, including splicing and a number of functional assessments, as well as strength assessments and quality of life measures as well. It's a 3-part study. There's a 6-month placebo-controlled multiple ascending dose portion, or MAD portion, a 6-month open label extension, and a 96-week long-term extension. Now, importantly, we've designed ACHIEVE to be something called adaptive in nature, which allows us to optimize the dose and dose regimen all within this single trial. And this is really part of our efforts to move as quickly as possible to advance Dyne 101. Our goal is to first show an improvement in splicing, and then over time to be able to correlate it to function. And that is what we believe, based on our conversation with global regulators, opens the door to potential expedited or faster approval pathways. Next slide, please. So on this slide, you can see some of the main inclusion and exclusion criteria. For the full list, please go to the ct.gov webpage or the uterusct webpage. And it also contains a list of sites for this global study. Next slide, please. Here I'll go into just a little bit more detail on the study design and just highlight a few things. First of all, the study is a multiple ascending dose study, as I mentioned. And here you can see that we are stepping through various cohorts from 1.8 milligrams per kilogram to 3.4 milligrams per kilogram dosed every 4 weeks and every 8 weeks, and then 5.4 milligrams per kilogram dosed every 8 weeks, and 6.8 milligrams per kilogram dosed every 8 weeks. And those are fully enrolled. So we're really excited about exploring the administration of Dyne 101 every 4 weeks or every 8 weeks, because based on our preclinical data, we feel that there is a very good data to explore relatively infrequent dosing. We do take a few biopsies in this study. We take 3 biopsies, one at baseline, one at 3 months, and one at 6 months. And these biopsies are critical to help us understand how much of the drug is getting into the muscle, and you'll see some of those results later, and what effect it's having on splicing. So that is why we take these biopsies. And then a really important feature of the study is that patients, after completing the 6-month placebo-controlled MAD portion, go into the open label extension and receive drug. So everybody coming out of the placebo-controlled portion after 6 months receives drug and are escalated to the highest tolerable dose. Next slide, please. I'll now start to get into the data. So we are very pleased with the favorable safety profile for Dyne 101 that we've observed and achieved as of the data cutoff date of May 8th, 2024. This is very differentiating from others in the field and speaks to the importance of each component of the FORCE platform. As I mentioned earlier, the Fab, which is smaller than the full monoclonal antibody and therefore makes it a bit easier to get into the cell, the clinically validated linker, and our choice of using an antisense oligonucleotide to target the nucleus, the nuclear toxic DMPK. Now, the safety data is based on participants, as you can see on this chart, dosed through multiple dose cohorts. During the MAD portion of the trial, most treatment-emergent adverse events were mild to moderate in severity and typically occurred around the time of infusion. There were 4 reported serious adverse events, both all deemed unrelated to study drug, and you can see them characterized here. One, as you can see, was a case of pneumonia. The other was a transient worsening of AV block in a participant with ongoing medical history of first-degree AV block. Unfortunately, the natural history of DM1 patients, as we've learned, there's a known background of arrhythmias. I'll also point out the liver enzyme elevations were seen in a percentage of patients, but no impact on liver function as measured by something called bilirubin, which is a way to measure liver function. There was no impact on that particular assessment. And unfortunately, hepatosteatosis is also known to be part of the natural history in myotonic dystrophy type 1 patients. Of note, there were no reports of anemia, thrombocytopenia, or kidney injury, and we've had a number of hundreds of doses administered in Achieve across the study, the MAD portion and the open label extension. And the profile has supported, the safety profile has supported the dose escalation you saw on the previous slide. Next slide, please. So one of the other aspects of what we're doing in our particular study is that trying to address the foundation of the symptoms seen in myotonic dystrophy. So we designed Dyne 101, as I mentioned, to address the spliceopathy aspect of myotonic dystrophy to enable comprehensive functional improvement. So as I talked about on the left-hand part of the slide, we have a targeted delivery mechanism so that the antisense oligonucleotide can attack the toxic nuclear DMPK and correct the missplicing or correct the spliceopathy. And we're able to measure this using a validated biomarker to measure splicing. I'll also show later a little bit of our data from our VHOT or myotonia data. And we know that this is an early indicator of functional improvement. So myotonia changes can be seen very early because of the pathophysiology of this disease. And ultimately, what's really critical is to show broad functional improvement in muscle strength, as well as measures of mobility and other function. And of course, we want to be able to show improvement in patient-reported outcomes as well. And so I'll talk a little bit about this data in just a moment. Next slide, please. Okay. So on this particular slide, I'll orient you to the slide in a second, but it's really showing how much of the drug is getting into the muscle and what effect this is having in terms of the splicing. So the gray bar is the placebo arm. The blue is the 1.8 milligram per kilogram cohort dosed every 4 weeks. The teal is a 3.4 milligram per kilogram cohort dosed every 4 weeks. And the dark blue is the 5.4 milligram per kilogram cohort dosed every 8 weeks. And what's nice here is you can see on the left-hand side of the slide, which is the concentration of the antisense oligo getting into the muscle. There's almost a doubling from 1.8 to 3.4 and a tripling from 3.4 milligrams per kilogram to 5.4 milligrams per kilogram. So we're seeing a rational increase in the concentration. On the right-hand side, you can see the dose-dependent splicing correction at 3 months. So in the 1.8 milligram per kilogram cohort, just every 4 weeks, there's a 13% improvement in splicing. So a decrease in splicing is an improvement. And 3 of the 5 participants in that cohort improved. 2 did not. In the 3.4 milligram per kilogram cohort, we had an improvement of 19% splicing, and every participant improved. And then in the dark blue, in the 5.4 milligram per kilogram dosed every 8 weeks, we saw a 27% improvement in splicing, which was really outstanding. So we have said for some time that based on our own preclinical work and input from others in the field and the natural history, that we believe 20 to 25% splicing correction is needed to drive functional benefit for patients. Because Dyne 101 targets the nucleus, where the missplicing manifests, We believe effectively knocking down nuclear DPK will drive that correction and splicing, as you can see here in the data, and lead to significant disease modification. And we believe we're beginning to see just that. Next slide, please. So here we show the results with respect to myotonia. And myotonia is the inability of muscles to appropriately relax after contraction. And we use something called the video hand opening time, or VHOT, to assess myotonia. This simply involves asking a participant to make a fist and then open it, and we time. So what we see is treatment with Dyne 101 demonstrated an improvement at all doses and time points. We see nearly a 4-second improvement at our lowest 1.8 milligram per kilogram dose at 6 months. At 5.4 milligram per kilogram, we see a 4.5-second change at just 3 months. As I mentioned earlier, the belief all along has been that myotonia may have the best opportunity to improve quickly with an effective therapy. So for this early read, we thought it was important to assess the VHOT, or the myotonia, and we were able to see robust results over a 6-month period. I will point out that you can see a clear worsening in the gray line, the placebo line. So these data are very striking, and we're excited to see how this and other functional measures evolve over time and at higher doses within ACHIEVE. Plus, we have additional data looking at measures of function and strength, including mobility assessments, on our website. And for even a deeper dive into the data, please go visit our website and you can look at the other data. But we also were very pleased to see trends of improvement in measures of strength and function. Next slide, please. So we're pleased to also share with you the patient-reported outcomes data. We looked at a measure called the MD-HI, the Myotonic Dystrophy Health Index. The MD-HI is comprised of multiple subscales, including fatigue, strength, GI disturbances, cognition, and the ability to do daily activities, amongst many other measures. So what we can see here is patients in the 1.8 and 3.4 milligram per kilogram dose cohorts experienced an overall improvement in the MD-HI and improvement in trends towards improvement or improvement in all 17 subscales. These PRO data are incredibly encouraging, and we look forward to further evaluating PROs as part of our commitment to comprehensively address symptoms of myotonic dystrophy type 1. Next slide, please. So in summary, we believe these data really demonstrate the potential of Dyne 101 in myotonic dystrophy type 1. We show dose-dependent muscle delivery and compelling splicing correction that was consistent across patients. We showed meaningful improvement in endpoints such as myotonia. And as I mentioned, we have also shown improvement in areas of muscle strength, time function tests, and patient-reported outcomes. What we see is a deepening of response with longer time on therapy and a favorable safety profile to date. We'll update our path to registration for Dyne 101 by the end of the year, and I look forward to coming back to this community and giving you another update in some months from now. Next slide, please. So with that, I just want to say thank you. We recognize the sacrifices that the families have made over the years, the investigators, the teams that are exploring new therapies. It is extraordinary what this community has done to advance therapies. And we know there's a significant burden that all of you take on to progress the science and advance the medicine. Thank you so much for that. And thank you to all of you for your time and attention today. And again, thank you to the MDF for having us here. And with that, Catherine and I will take questions.
Thank you so much. That was fantastic. We do have quite a few questions that have come in, But I would like to remind folks, if you have questions now after this presentation, that you can click the Q&A button to open up the question window. So just type your question in to that text field, and we will see those questions, and just make sure to click send. All right, we have quite a few that have come in. We will start with some very straightforward questions. Where are your trial sites in the US, and how can someone sign up just to be notified if trial sites will open near them?
Great. So currently, all of our sites are in Europe and New Zealand. We are working with the FDA to determine how to bring the trial to the United States. And again, I hope to have some more information for the community closer to the end of the year. But it is not lost on us at all that right now there are no sites in the United States. We are in close contact with many of the key opinion leaders in the United States. They're aware of the program, of the data. We're always in touch with them. And just like us and you, there is a lot of eagerness for us to be in the United States with this trial. So I can't give you much more information at this time other than we're working with regulators to bring the trial to the US. And we definitely know that there's urgency around this, as we've heard from you and investigators, and we feel ourselves. I don't know, Catherine, if you want to add anything.
No, just well said, Ash. I think, we do hear this question a lot, and we are very aware that we're, educating and working very closely with the community in which right now does not have access to the trials in the United States.
Yeah.
Great. Thank you. We have a question related to DM2. Can this approach treat DM2? Can the FORCE platform be used for other repeat expansion diseases? And if you could expand a little bit just to help folks understand, like, what is different about DM2 than DM1 in the starting point for many of our companies that are starting with DM1, and folks trying to sort of understand a little bit more about what makes them different and why the approaches may or may not have to be different.
Yeah. So I'll assume that the community is very aware of the differences, but essentially it's a different genetic basis of the disease. So DM2 is related to CCTG repeats, and DM1 CTG repeats. So it's a different target, if you will. So the FORCE platform is a platform that we have now shown across different neuromuscular conditions to be able to effectively deliver payloads for whether it's a disease where there's a, like, myotonic dystrophy, where it's a toxic gain of function, if you will, or even Duchenne, which is a toxic loss of function. So the FORCE platform can apply to DM2. We haven't, done a lot around that internally yet in terms of the research, but theoretically, the FORCE platform could be used for DM2. Sorry, was there a third part to that question?
It was basically just, it appears that folks are tending to start with DM1.
Sorry.
Yes. So just, what might make them different, or why might DM1 be the place to start? And then how do we know if we can actually expand to DM2?
Yeah, great question. So the simplest answer is that the prevalence of DM1 is much greater. And so I'll try to sort of maybe break it down into a couple of things. One is the number of patients with DM1 is much, much higher than the number of patients with DM2, much greater percentage. So to be able to actually do a trial to show your platform works in a condition like this, it is more efficient, faster to be able to do a trial in DM1 than DM2 because of the availability of a population with DM1. Not minimizing DM2 at all. It's just we're trying to move fast to show that this platform works. And hopefully it does. to date, it seems like it has. I'll also add that the FDA has recently come out with something called a platform technology guidance. And so what's nice is the FDA, it appears, is opening up the ability to have a dialogue, to be able to think about how to more efficiently get drugs through the system for approval when using a particular platform. And so we have our FORCE platform, and we think this guidance would be quite useful in having those discussions with the FDA. But right now, this is our first trial, and we're trying to make sure that we're able to do what the platform needs to do. And then that opens up many doors for us. I hope that helps. Catherine, you might have better explanations, but hopefully that answers the question.
Thank you. That was very helpful. A question came through asking how you really know you're hitting your target if this is a multi-systemic disease. How are you hitting your target and ensuring that there aren't other negative side effects or effects in general for other systems that you're not specifically targeting? So how can you accurately monitor all of the body systems affected by myotonic dystrophy?
Yeah, that's a great question. So some of the question will be specific to DM1. Others are just related to drug development. Another rating is just related to drug development. So how do we know we're having on-target effects first? So we're measuring that. So, as I mentioned, we take the biopsy and we take baseline and then take it during the trial. So we're able to show that we're hitting our target of DMPK, knocking it down, that we are actually affecting this very important foundational aspect of the disease, the splicing. And it's well characterized in the literature in terms of how to analyze missplicing. And we're working with, Professor Charles Thornton, who's a luminary in this field, to help us make sure that we are looking at splicing accurately. Then, are we having on-target effects in terms of, are we doing something meaningful from a functional point of view? So we talked about myotonia, and that's an early target, early indicator. What I didn't show today is the data that we have shown back in May, which is improvement in QMT, so muscle strength, improvement in time function tests. So we are also showing improvements in our study with Dyne 101 that one would expect if you are actually able to correct the splicing defect. So we're really pleased to see that. And I talked about the quality of life pieces as well. Now, the question I think is also, how do you know you're sort of getting at things comprehensively? That's a tough question, but it's really important. So the MD High, for example, has 17 subscales. And some of the subscales are fatigue, cognition, communication, GI disturbances, sleepiness. And so these subscales have components that are driven by the brain, sort of central nervous system driven, and skeletal driven, probably a combination of the two. And so we are triangulating with as many measures as we have at our disposal that we're able to affect, the various symptoms that we talked about earlier in the presentation. So that's one piece of on-target comprehensive care and effect. How do you know you're not doing something untoward? How do you know you're not hurting some aspect of what's happening? So the protocol is constructed very carefully on the back of so much preclinical data. So we used Professor Thornton's HSALR mouse model. We developed a mouse model where we can actually test our clinical candidate in the mouse model because it expressed the human transferrin receptor and the human toxic DMPK. We assessed this drug in multiple monkey studies as well. And that generates a great deal of information with respect to safety and tolerability. And so that information that helps drive what our protocol looks like, not just in terms of dose and dose regimen, but also what are we looking at from a safety profile? What safety parameters do we need to include in the protocol? So we talked about iron transport, right? If for the TFR1, its normal function is iron transport. So we're looking at measures to make sure we're not disrupting iron transport. So all of the information we gather in terms of the mechanism of action knowledge, the preclinical data, all of that feeds into constructing a protocol that is very carefully monitoring safety across every organ, across the entire body. And there's a very, that's of paramount importance, especially for this phase 1/2 study, which is a safety and tolerability study. So we're looking at probably every safety measure that one can imagine to make sure we're not doing something that we shouldn't be doing.
Thank you. There's the sort of follow-up question that came through directly that asks if anyone has dropped out of the trial due to side effects.
I think as of the data cutoff date, no. But if I am wrong, Tanya, we will submit a correction. So I apologize, but I just because I think as of the cutoff date, no.
Okay, great, thank you. Will Dyne 101 help with brain fog or reverse cognitive effects? So some of us have talked with neurologists who tell us that myotonic dystrophy does not affect the brain, but we know for sure that it does. So how does Dyne 101 impact the central nervous system?
Yeah. So I'll answer in a second. So my apologies. One participant did withdraw from the study as of the data cutoff date. So sorry about that. Yeah.
Thank you.
CNS. I think your question was around CNS. So the R receptor was designed to be able to target muscle. Our antibody was designed to target the TFR1 receptor and bind to with an affinity so that we could drive it into cardiac, smooth, and skeletal muscle. And we're seeing that we're doing that. Now, what I have shown in the past is with a tool compound that our conjugate is able to get into the brain. So we did studies in non-human primates or monkeys where we delivered systemically, so intravenously, our conjugate. And we showed that we could not only get into the superficial layer, so the cortex, but also the deep brain, the caudate and the putamen. So we had wide distribution at the superficial and deep brain level in monkeys. And this is really exciting. This is really, really exciting because we continue to try and explore what Dyne 101 or what our conjugate can do. And so this tool compound, which is similar, gets into the brain very, very well. And we did an interesting experiment. We actually compared it to the unconjugated ASO delivered IV. And it didn't penetrate it didn't get in the brain, which is to be expected. And then we also compared it to something called intrathecal delivery of the antisense oligo. And it showed that just was distributed in the superficial layer of the cortex, but didn't get in the deep brain. So we're really excited that we're able to cross the blood-brain barrier and get into many components of the brain. And then we did an experiment in that mouse that I was talking about. And we actually showed in the cortex and the cerebellum a decrease of those trapped proteins. So it was really, really exciting work for us to say, okay, in the monkeys, we're getting in there. And in the mouse model that's relevant, it's a relevant mouse model, we're actually able to have something called a pharmacodynamic effect by reducing that trapped foci. So super exciting. And then, as I was alluding to the MD high, I was pointing to certain subscales that indicate we're actually having some effect in the CNS. But now as we move forward in the development program, we're going to hone in on how to answer some of these questions in our phase 3 program. Maybe there are specific measures that will help us better understand our ability to affect the CNS. So we're exploring that and working through that, working with experts around the world.
That's exciting. Oh, okay. You've shown a 27% improvement in splicing. Does that mean that 27% of the trapped proteins in the hairpin loops are actually being released? How can you define this a little bit more?
Sure. Yeah, so I think that's a long answer. I probably wouldn't do it nearly the justice. I know I wouldn't do it nearly the justice that Dr. Thornton would do. But basically, there are thousands of splicing factors, and over the years, there are a handful, and one can debate if it's 10 or 20 or 22 or 30, but 22. There's a 22 gene splicing panel that is widely accepted as being able to be correlated with functional outcomes. And so the natural history work done from NDM1 points to this 22 gene panel correlated to changes, to correlate to functional outcomes. And so we are using that 22 gene panel as a biomarker, if you will, that should be predictive of clinical benefit. There are really great papers out there for who's interested in going deep into the derivation of that 22 gene panel, and people can go back to Dr. Thornton or Dr. Wang or others. But I would be the wrong person to dive deep into the derivation of the gene panel. You wouldn't want me to do that.
Thank you. Okay. Next question. It appears that the splicing improvements are higher in the 4.5 dosage every 8 weeks as compared with the 3.4 every 4 weeks, even though overall less medication is given with the 4.5 than with the 3.4. Why is that?
Yeah, no, it's a great question, and I should have explained it a little bit better. So, so I think they were saying 4. So it's the 5.4 milli. So it's 1.8 mg per kg every 4 weeks, 3.4 mg per kg every 4 weeks, and 5.4 every 8 weeks. So great question. Why is that less frequent? Why is that higher dose less frequently leading to better splicing? So the first 3 months of the administration of that Q8 weekly, of that every other month cohort, is actually given every month. So 5.4 is given every month for the first 3 months. Then it goes to every 8 months. So in a sense, it is apples to apples comparison here in terms of the data. That's the cohort is technically 5.84 every 8 weeks, but the first 3 doses are given every month.
And will that dosage or frequency of administration change at all if you get to, hopefully, phase 3? Do you anticipate anything at this?
So can't predict, right? We're still in that, this year is still about nailing down the dose and dose regimen. So we're still exploring what this is going to look like.
Okay. I'm not sure I'm fully clear on this, but maybe can you explain registration a bit more? What does it mean that you can go to the FDA for approval by the end of the year?
Sorry, we're not I'm not saying we'll go to the FDA for approval by the end of the year. We are going to continue to have dialogue with the FDA and then hopefully provide another update before the year is over. But we're not going to the FDA for approval. Okay. So registration just means we're designing a study that could potentially allow us to have a dialogue with the FDA, depending on the results of that study and, of course, how it's designed, to discuss, is this something that would be acceptable for registration for ultimate approval? And again, splicing appears to be a good biomarker for that.
Okay, great. Well, there's a follow-up question that's asking sort of what's preventing the FDA from approving trials in the U.S.? Do you have any sense about that?
Yeah. So again, I want to be super clear. I am not at all suggesting that the FDA is not approving trials in the U.S. That is not at all what I'm suggesting. We haven't filed our IND in the U.S. Because we want to be able to be in the best position possible to have a dialogue with the FDA and start trials in the U.S. at doses that are going to be impactful and meaningful. So, it is, the FDA has not said to us, we can't start. And we know that the FDA allows many sponsors in many different indications to do studies in the U.S. So that is, they look. The FDA is, I think patient safety is paramount, and we're very fortunate to have this organization, work with us, work with sponsors, work with academics and advocacy organizations, because I truly believe they want the same thing we do, transformative therapies that are safe. And I believe that of the FDA, and I believe that of payers, insurance companies around the world as well. So insurance companies and payers around the world. I think we all have the same goal, and just we try to do it in the way that we're supposed to be doing it. So
that's really helpful. Thank you, because I know a lot of folks are wondering in general for the companies that aren't launching trials here in the U.S. Is it because the FDA is preventing it, and is there something that we could do or that the community could do? But in fact, you haven't actually gone through that process, so that's really helpful to know, and I hope that helps answer the question for a lot of folks who are wondering what might happen there. So I appreciate it.
May I make one comment? I think, or maybe Catherine, you'll want to make a comment on this as well. I do think, as with any community, the voice is so strong, whether it's myotonic, whether it's ALS, whether it's MS. So the FDA, in my estimation, is incredibly open to hearing what's important from the patient community, and your voice carries tremendous weight.
Thanks, Ash. I was going to pick up on exactly the same thing, because I think there's the question about what is Dyne doing, and the question of what can the patient community be doing, and to that and they are very clearly interested in hearing from the patient community around what is the burden of disease and what is the unmet need. And, the more they hear about the urgent need, I think the more myotonic dystrophy certainly elevates its presence to them.
Sure. Thank you. Question about if the drug is working, if someone's actually going through the trial and they're a participant and the drug appears to have an impact for them, will they be able to keep taking that drug if it's working for them?
I'm just trying to understand it. So I assume they're saying post trial completion, I guess is what they're asking. Okay, got it. So the trial is 3 years in duration, as I mentioned. We will not allow a participant in our trial to be without drug after the trial is over, should he or she want to stay on that drug. So, as long as the family, that's the participant, and their physician would like that participant to stay on drug post the trial, we will provide drug for that patient.
It's helpful. Couple of questions about just sort of trials and costs. So when it would be available. So at this timeline right now, if you're looking, hopefully moving to a phase 3 in the near future, once you have your results, how long would it take, timeline-wise, to just get this drug to market and make it available to folks? And the second part of that would be, how do we know what it's going to cost? Are we going to be able to afford this drug, and our insurance cover it? And I know you don't have the answers to all of those questions, but if you had a crystal ball, or if you could give some insight to that, what might you say?
Yeah. So timing. Let's talk about timing. I think that we are, as I mentioned, still exploring the dose and trying to hone in on the dose and dose regimen for phase 3. So we do. I mean, as I'm sure you would imagine, we have our own internal timelines that we are aggressive internal timelines for completing the development program and filing for approval. So absolutely, we have timelines internally. We don't share those timelines right now because it's so dependent on so much. It's dependent on the results we're getting as I speak. It's dependent on the conversations with the FDA, with regulators around the globe. And so, it wouldn't be, it's not the time to share like details around timing because we have so much that we need to work through that, is with respect to the trial conduct and then regulatory interactions. And then on top of that, going to the next part of your question, is there's the reimbursement piece. And so we need to then have conversations with insurance companies, payers outside of the US. There's a number of single payer systems and other complicated systems outside of the US that all have different requirements. And so, we also have to look at and I'm not trying to avoid the question by any means. I'm just trying to give a little bit of insight. So how we think about the value of the drug, we gotta show that the drug works really, really well, that it is disease modifying, that it's delivering an efficacy and safety profile that not only physicians value and regulators, but that payers value. And so you're going to have a lot of conversations with payers to talk about, here's the data, here's how it's changing lives, here's the data we're seeing, in the short term and in the long term. And you discuss this with payers to get to a point where you're able to actually come to a common place on the value of your agent. So can't give you much more than that. But maybe Catherine, you want to add to it?
It's a tough question.
It's a tough question
to answer at this point. Yeah. Well, we will.
Maybe I will say, yeah. I'm sorry. Go ahead.
No, I was going to say the foundation will certainly work on that and talk with payers and sort of do everything we can to ensure that this will become any drug that is approved for myotonic dystrophy will become available and will reduce as many barriers to use as possible.
Yeah. I'm glad you said that. I was just about to say something similar. I think that would be amazing because one of the things that we're probably going to face is a lack of understanding of the disease of myotonic dystrophy with payers. And so, how can we partner together to be able to help the insurers, the Anthems, and the United and Aetnas, and all of the systems in the U.S., outside of the U.S., understand the disease, the natural history, and the outcomes that are most meaningful for families. And that is going to be a tremendous amount of work, and that adds so much value and speed to the entire process. So just a thought that we've talked a lot about at Dyne.
Yeah. You have very specific inclusion-exclusion criteria right now.
Like?
At what point might that change? So when would we know that this would be acceptable for children or for people who are outside of your inclusion criteria at this point?
Yeah. So this is the phase 1/2 study, primarily looking at safety and tolerability with the other measures that I mentioned. The regulators require us to go into adults first for phase 1/2. And so that's what we did. We do plan to think about how to have a broader inclusion-exclusion criteria as we get into later stages of development. So typical drug development is in your phase 1/2, you're very narrow in your patient population. You have very strict inclusion-exclusion criteria. You try to create as homogeneous a population as possible so you can be really clear about your safety and early signals of efficacy. Excuse me. And then as you move forward in your development program, then you start to think more expansively about the broader population for this particular disease. But we do need to show safety and efficacy in adults before we can go into children. We absolutely do. And then we also think about what outcomes measures we need for a younger population because they may be very different from what we're using in the adult population. But we first have to show what we're doing in this population to then think about going into an earlier group. That said, at Dyne, we are thinking about this already. We are starting to put pen to paper about going into a younger population, but we really need more information from this study to solidify that path.
That makes sense. Thank you for that. We're a little over time, so I want to.
Oh, sorry.
No, that's great. There are a lot of questions, and so we appreciate that and want to make sure we can just answer a couple more, and then we will sign off for the day. But one question comes through: when will you know the impact of reducing expression of normal DMPK?
So there is no ability to measure normal versus toxic DMPK in humans. The assays exist, like we can do it with the mice, but the amount of tissue that we take in the biopsies doesn't allow us to do that. So we look at total DMPK knockdown. So when the question comes, like, is there a negative impact to reducing wild type DMPK? Again, this comes back to how our protocol is constructed, what safety parameters we have in our protocol to be able to make sure we're not doing anything untoward. But we can't tease out wild type versus toxic DMPK in humans with the amount of tissue from our biopsy.
Okay, thank you. Do the symptoms of DM1 occur due to proteins being bound in hairpins in the nucleus, so they're unable to perform their normal functions, for example, as amino acids as part of the normal chemical pathways in the body? Or are the symptoms due more to the hairpins in the cells blocking otherwise normal cell function due to their physical size?
So I probably would need to hear that like one more time, but let me see if I can answer the question. So we have these hairpin loops that, you described, Tanya, that I described, that are trapping splicing factors in the nucleus. So, right. So, if you imagine that hairpin loop, there's like little bulbs on it, right? These are the splicing factors that are trapped on that and can't get out. They need to get out so they can do their work, their cutting and pasting work, so that they can cut out those introns, get the exon squished together, and form the proteins for normal function. So it's these trapped proteins. It's these trapped proteins on that loop that are problematic. They need to be freed so that they can do their splicing job and create the proteins that are needed. And if you can't do that, it leads to all of those clinical consequences that we talked about earlier. So I hope that generally answers the question.
Oh, I hope so for that person. Okay, we just have a couple more.
That's what you said. That's what you just said. You said there were a couple notes.
No, we're close. We're close. I promise.
I'm kidding. No, I'm kidding. I'm happy to stay on as long as you want.
Really just a couple more that are a couple related, but one is going back to the 27% reduction in the CASI score. That looks to be the mean. What is the range from least improved to most improved for that 6 out of 6 patients?
That is a really important, good question. I should have had that number with me. I don't have the range in my mind, but I think it's a great question because you don't want to have like this, tremendous variance range. I will. So I apologize. I don't have that number. What I will point out again is that every patient was a responder. So all 6 improved in their splicing score. But I understand what's behind that question. So it's a great question.
Thank you. And then really sort of the final question is, how can people be notified if trial sites open here? And then if they would like for their local hospital or clinic to become consider becoming a trial site, who would they talk to or how would they go about that?
Yeah. So I'll answer, and maybe Catherine, you may have you may want to add. So I don't think this is exactly what they're looking for, but there are obviously alerts on ct.gov that you can you can have. So if there's an update to rct.gov page, you can be notified automatically. So that's just sort of, a mechanism to stay updated. And we update our page with I won't say there's not like a regular frequency, but whenever there's important updates, we update it, and then that alert can come through. The other piece of this is we have a clinical trials mailbox as well. And Tanya, offline, we can send that email address to you, where people can inquire about the status of our clinical trials and sites and such. And then finally, not to put too much on Catherine, but I think the last slide had her email. You can reach out to Catherine, and she can provide you information as well. It's really important that you feel like, and because we have someone that you can reach out to, and in terms of Catherine, and so happy to give you more information as well.
Yep. And I did put, so for the attendee who asked that question, I did put the clinicaltrials.gov link into the Q&A box, as well as my email address. So two very good sources of information. Thank you.
And then, the question about how do they get their physician or their clinic part of our clinical trials? I think it's a great question. It's a discussion they can have with that physician, and that physician can always contact, again, use the same mailbox. Frankly, can contact me directly, can contact Catherine, and then we can have a dialogue. It happens not like often, but it does happen from time to time when physicians reach out to us either on their own or as prompts from patients that they're seeing, and we're always happy to engage with the physician if they're interested in participating in our trial.
Also, the Myotonic Dystrophy Foundation, right? We will put out any changes usually in our dispatch, so you can look for sort of updates and when there's new data, and we're happy to share that all with the community. And if folks have questions and they can't get a hold of anyone, please do contact us so we can make sure that we connect you and get you the information you need. So I want to thank you, both of you, for the presentation today. This was really helpful, and we're incredibly hopeful at this point. We look forward to hearing more about your progress very soon. I think this is the end of our webinar. So if you all in the audience have liked today's webinar, please consider donating and helping us to further support finding a cure. Also, please consider joining the registry if you haven't already. The registry is extremely important, supporting clinical trial activities, and it is one way that we communicate with folks right away when there are changes to sites, or if you might be eligible to participate, or at least considered for a particular trial. So without our registries, we wouldn't be able to connect you with these trials and trial centers as fast as we would otherwise. So please do sign up. We are grateful to Dyne. Thank you for the work you are doing for our community. Thank you for taking time with us today. And to everyone else, have a fantastic weekend. Thanks, everyone.